Okay, I guess we'll just make a start now. We've given everyone the obligatory 5-minute academic late call. So again, thank you for joining our webinar. I'm Jessica, and I am a project manager at the UKCRC Tissue Directory and Coordination Center. As our name implies, we're the organization that has developed the Tissue Directory, the topic of today's webinar. We'll be recording this meeting, and so the demonstrations, polls, or anything typed in the group chat that you'll see at the bottom of your screen will be recorded in a video and made public on our website for posterity and reference. So, for example, if any of your colleagues couldn't have made it today, they can access the video and watch the demonstration.
So I talk you through the main parts of the tissue directory, mainly how you register there. I'm on the test system, so don't use this URL because you won't get through to it. So the main one, we've got some of the web links here. Just to freak you all out a bit, when I click this, it should open up the tissue directory for real for you. So that's the main URL for us, is the directory.biobanking.uk.org. So that's where to go to. What I'm going to talk you through is the main registration. I've logged in already, so I can go to my Biobank admin tab. So on here, you can see everything that you've put in so far. So usually when you first come in to register, you'll just be here. So this will give some information about the biobank that you're wanting to put in. And if you want to edit it, you can come here. So most of this information should be relatively straightforward. And we might do another poll in a second as well about just understanding how far you've got in the registration process. Because some of you may have got this far, some of you may not have got this far at the moment. So on here, we've got the name of the biobank, which you should understand. Description. URL is if you've got a website you want to go to. Contact is something quite important. So this isn't the email or your personal email address, but the email address you want any contact from researchers to go to. So this will capture any communications will come in via this email address. So you can have one email address for your login, but this can be a separate one where you direct everything through to. The logo is then if you've got your particular logo, address, city, country, postcode should be self-explanatory. Then what we do start to have under governance is the institution, school, department where the biobank resides. And we also capture now the ethics and HTA license numbers. So this is because we are working very closely with them. And as we said, we've been working on them particularly around the ethics. So when you apply for a research tissue bank status or renew for a research tissue bank status, it will now be a condition on your ethics that you register in the directory. So to capture this number just allows us to feed that information back through to them. With the services, this is if you offer additional services, so you can kind of tick the ones that apply. This list is not exhausted at the moment, so if you see services that aren't there that you want to be added, just let us know and we can add those in for you. Data sharing is then something quite important. So one of the things we want to do with the directory is make sure that we have the information come through and register with us once, and then we'll share that information as much as possible. So for example, we mentioned already BBMRI, and if you register with us, we'll then make sure your profile goes through to BBMRI ERICS directory, which is a European-wide one. If you clicked opt out, that will ensure that none of your data goes anywhere apart from within the UK directory. So that's something that you've got. So we will do the second poll now just to see where you've got to. So if that can be brought up, that'd be great. So this is just to give us an idea of how far, if at all, you've got within the registration process. So I think 3 out of 4 were involved in biobanking. So yeah, there's a good response in here. So aware of the directory but not registered, hopefully you will then go on to register today once we've shown you how to do it. And those of you who have got the basic information in, we're going to go into the details of how you add in the extra information to your profile to make sure it's fully discovered on the search system. So one thing I will mention, once you've now done this part of the process, your biobank will then become listed on our A to Z. I'm just going to bring up our main directory page again. So you'll see here we've got a search by disease, but we've also got an A to Z on the right. So the A to Z of biobanks is once you've completed your basic profile, you will then appear on our A to Z. The search by disease and the other categories I'll go to show you in a second, but that's once you've added in more details. So at the very start, you'll get this basic information come through about the biobanks in the A to Z. So first going to talk you through some of the main concepts. So we have collections and capabilities. So collections refer to samples that are already in your freezers that you've already collected, and capabilities, it gives you the option there to add information about samples that you can then collect. So it's the collections, historical, capabilities, what you could collect in the future. So what I'm going to do is add in a new collection just to show you how you can do it. So the disease status is based on SNOMED. So as you start typing, you'll get a list of conditions that appear. There's also other ones which are non-cancer. So you can just start typing the term that you think it is, and they'll pop up. What we haven't done is added all the terms of SNOMED into the directory. This is because we wanted to try and keep some control over that, but also not to confuse you with various levels of detail that can appear via SNOMED. So if the term isn't present, get in contact with us because we can then add that in very simply. And we usually have a conversation with you about the best way. So there is this on one of the links here, the all SNOMED terms. Have a look at that. That will take you through to a browser of the entire SNOMED, and that will help you see which terms you may want to add. We can also help you with that as well. And more generally, there's some help sections available on our website, so help pages for registering, and our YouTube channel. Check these out because they've got some videos as well from before. So if I continue now registering this, I can put a disease status in. Title is something completely optional. What people have used this for is if it's a clinical trial, for example, and it's got a particular trial name, or if you have got a particular name for the collection. But for many, this is nothing that's very important. Description should be self-explanatory again. You can all see how good I am at typing today, which would be cool. So you can enter the description there, and then the year that it started. And if it has finished, you can put the year it's finished, but if not, obviously don't worry about it. That's an optional field. Access conditions. So this is trying to understand how people can get access to your samples. The directory itself and the center does not get involved in any of the access decisions. That is still completely under your remit. We don't get involved in that. All we want to do is give the researcher who is searching the directory a fair appreciation of what they can expect. So for access, it's kind of open to applicants, which means you open and anybody can apply. And there's various degrees of open. So you might only have particular calls or only via kind of more research collaboration, right the way through to kind of closed to access currently. So because there's a requirement on you to register in the directory as part of your ethics permissions, we didn't want to close the door to people that had different access criteria. So just put the one in there that's most relevant. So this is kind of you select one as you go. Collection type is something that's really come through from the MyAbyss data standard, which is a European data standard. Again, these might not fit you exactly, and if there's a real problem getting contact, we can add a new category in, but it's selecting the one that's most relevant to you. I should also say, for those, there are definitions on our help pages. There's definitions for each one of those. The collection status, again, should be self-explanatory, and the collection point also. So this is trying to give the researcher understanding of when the samples were collected in relation to the diagnosis that we've just put up at the top. So was it pre, post, during, or have you actually got samples at multiple time points throughout the process? So you, again, select the one that's most relevant. We also understand that people will have different consent forms with different potential restrictions on them. So if there are restrictions such as not being able to supply samples to any commercial organization, tick the ones that are relevant to you. Associated data is just trying to get us to give the researcher again an understanding of what data you have available on those samples. So we've got the list here, so you have to tick both the type of data that you have and how long it takes you to provide it. If you just select the time frame, it will automatically select that category for you. So again, you don't have to select any of these. This is an optional field in that sense, but it's just selecting the ones that are most relevant to your collection. So by immediately, what we mean is that you have that data readily to hand, that if somebody came to you with a question, you could answer that question essentially very quickly, versus actually it might take you up to 3 months, 6 months, or more than 6 months to answer that question. So it's trying to give the researcher an understanding there. So that's the highest level of what we have in terms of the samples in your freezer. That's called the collection. We now drop down to information about what we call a sample set. Now, what this starts to do is understand your collection, but broken down by gender and age. So we ask you to enter in separate information for each time there's a different gender and a different age. So here we can enter male, we can enter the age that it's relevant for, and then the number of donors. So as you'll see, as I move this box, it's bracketed, it's not exact, and the idea here is not for us to kind of pick you up if you said 100 to 500 and actually you've got 510. It's just to give the researcher an understanding about what level of samples or the number of donors you have in your biobank for this collection based on this gender and age. So it's an approximation in many ways, and select the one that's most relevant to you. The next thing we ask is the types of samples that you have available. So you could say whether it's tissue, the temperature it's stored at, and roughly what percentage of the donors do you get that tissue on? So is it on something around 10%, or do you usually get this on between 50 and 75% of the donors? The macroscopic assessment is understanding in the disease that we've selected for the collection, are these samples affected by that disease or not? And obviously in some situations that would be not applicable, but you select the one that's most relevant. Now what I'm going to show you next is some of the copy and paste features that we have, because obviously if you're having to enter this information in per gender, age and sample type, it starts to get a little bit laborious. But what you can do is hit copy. So once you hit copy, it will bring up the information you entered before for the tissue, and you can now change this over for the other sample type. So you can keep going with this copy procedure until you've got everything in there that's then right. So now I've entered that information in that sample set for males who are adult. If I want to add in for females with exactly the same information, we then have another copy facility. So you click on here, go to copy, change it to female, and that's added all that information in exactly the same for male and female. So our advice is to add one in right for that particular gender and that age range, and then you can copy it between the different ones to try and speed data entry. And this completes then the data entry for the collections. So you've now entered information about diabetes, when it was collected, and the samples that you have available. And at any point, you can come back and do this. So on our directory, once you've got to that level of information, you would then appear in this list. So if I now type what I'm searching for, you will appear on these search results. Whereas until you've done that last part of adding the sample sets, you won't appear here. So that's one way of checking that you've got the data right. If you're not appearing on this part here, it's most likely because you haven't entered in sufficient information in the sample set. If you're confused about that at all, give us a shout and we can help you as well. The other thing to show you is capability. So capability refers to samples that you can collect in the future for somebody. So if I add a capability in, this one's a lot quicker. Again, I'll just do diabetes. We'll then ask you the protocols. So can you collect these samples on a bespoke consent form, so a consent form provided to you by the researcher, and are you able to collect these samples to an SOP that should be provided by the researcher? Again, you tick the ones that are relevant, but you don't have to select any. We want to then give them an idea of how many donors do you think you'll get in a year for this particular disease, so you can enter in a number here. And it provides, before, exactly the same information about data. What data sets do you have readily available that you could access immediately, such as an electronic health record, or which data sets would take you a little bit of time to actually go and collect for them. And that's you added in the capability. So from that side of things, capabilities are much quicker and collections take you slightly longer, but they're both relatively simple. Collections also, if you need, we've got a bulk upload. So if we can send you a spreadsheet template if you've got multiple diseases, and we can show you how to do that. The other quick functions I'm going to show you just now is the admin. So this is, at the moment, I'm the only person that can edit this profile via my login. But if you want to, you can add a name and email address of other people you want to be able to access this. This then comes completely under your control. You can control who can have access and who can't. So you can do that now. Okay, so funders is the last one I'm going to show you. Some of the funders want us to be able to track who's registered and who's not. So here you could add the funders that kind of currently support you. So I think this is a drop down again. So you start typing and it'll start adding those to the list. So if you haven't got the funder in the list that's there, we can supply those through to you as well. So I think that's really it from the directory perspective. We can show you the search as well if people are interested, but from a registration point of view, that's very much it. So I think we can open up to any questions and discussion points. If you want us to go through anything more in detail that you've struggled with, for those that have entered the basic information but not got any further, or those who've created the account not get any further, we can do that now as well for you. So please ask away. I will also stop the recording now so that we can do this without broadcasting this to the world.